this so correct and thorough presentation of me. So thank you so much. And as you mentioned, yeah, I'm going to talk about microeukaryotes within the microbiome of marine animals. And I know you already had many guests, and probably you already know what a microeukaryote is, but I would like to start. I always start with this first first slide. This is the tree of the eukaryotic life. Okay, and I use it as a map. Can you see my pointer? Perfect.
Yes.
So I use it as a map. We are here, so within the metazoans. Our mushrooms and other fungi are here, and our plants are here. And these are the three most charismatic eukaryotes that happen to be the three big multicellular eukaryotic groups. However, they only represent a fraction of all the eukaryotic diversity, a tiny fraction. And all the other eukaryotic diversity, they are what we call protists. And most of them are unicellular, and most of them are microscopic. Okay, and these are the main groups of eukaryotes. Here we have the amebozoans that include well-known parasites like acanthamoeba. Here we have the excavates as well, contain some parasites like giardia, the archaeplastida that include the plants and most of the algae that we know. Rhizaria that include the beautiful radiolarians, alveolates that include the dinoflagellates, but they also include the malaria agent. Estromenopiles that include the diatoms and also include many other tiny little flagellates, heterotrophic flagellates called masts that are very abundant in the ocean. And here we have the Opisthoconda that include us and all the other animals, include also the fungi and a diverse group of unicellular organisms related to both fungi and the animals. So this is the landscape of the eukaryotic diversity that is heavily dominated by unicellular microscopic organisms. And microeukaryotes are not only important because they are diverse and because they are the most diverse eukaryotes, but also because they play different roles in marine ecosystems and also in other ecosystems, but I'm going to be focusing on marine ecosystems during this talk. Here you can see that in the ocean, microeukaryotes can play the role of photoautotrophs, but they can also be saprotrophs, so heterotrophs. They can be parasites, they can be predators, they can be symbionts of other organisms like animals, and also they can be mixotrophs. So that means that they have this dual behavior of being phototrophs and being heterotrophs, depending on the situation. As well, animals are important for health. For example, for animals, sorry. Microeukaryotes are important for animal health. As you can see here, this is a pie chart that reflects the World Organization for Animal Health notifiable diseases. 10% of them are, 15% of them are caused by protists, and most of them are caused by bacteria. Among the ones caused by eukaryotes, protists are the ones that dominate. In the case of agents responsible for extinctions and extirpations, that means or complete extinctions of animals from Earth, or the local extinction, so extirpations of animals for certain regions of the Earth, protists are responsible for 25% of these extinctions and extirpations. For all the others, I mean, most of them are fungi, and many of these fungi are also unicellular eukaryotes, okay, are not multicellular fungi. So microeukaryotes are diverse. Microeukaryotes are relevant in the environment, and they are also relevant for the economy, for health, and for conservation. Okay. So let's talk about microeukaryotes that are associated to animals. These organisms, as bacteria do, they can play three different roles main roles as symbionts of animals. They can be mutualists, they can be commensals, and they can be parasites. Now I'm going to ask a question. So do I need to explain these concepts? No. So let's, okay. Classically, the view we had pre-microbiome era for all types of microorganisms is that those organisms associated to animal hosts, most of them were parasites. So they were pathogens, they caused disease, they were essentially bad. Some of them were mutualists, symbionts. One example would be the zooxanthellae in corals, and just a fraction of them were considered to be commensals. As I said, this is the view we had pre-microbiome. Okay, this is an example of different eukaryotic organisms that we can find in the human gut. So in order to differentiate what we look at from what people working on bacteria look at, we coined this word eukaryote. It's not a very nice word, but it's necessary because despite they are part of the microbiome, when you say microbiome, by some reason, people, they only think about bacteria. And then we use this concept eukaryote to refer to the microeukaryotes within the microbiome. People working on bacteria are lucky they don't need to use the word bacterium because, I mean, if they say microbiome, everybody will know that they are talking about bacteria. So these are some examples of microeukaryotic members of the microbiome. So as I said, this is the view we had of the microbiome before of the microorganisms living associated to animals before the microbiome era. After the microbiome era, and that means since we started to use molecular techniques to describe the microbial communities living within animals, living within humans, the picture we have is this. So most of the microbes living within a host, they are commensals. They are just there. They are not parasites. They are not mutualists. They just live inside the animal or on the skin or whatever, because that is their environment, and they don't do any harm and they don't provide any benefit to the host. As I said, these are the majority of the microorganisms living associated to a multicellular organism. However, those are not the ones that we study the most. In the pre-microbiome era, and the reason why we had this idea that most of the microorganisms living associated to a host were all parasitic and some were mutualistic is because most of the research was targeted and was based on microscopy and isolation of things that we were looking for. And understandably, because we are humans and because we care about ourselves, we care about our animals and our crops, we had a strong interest to study parasites because they harm us. So we targeted parasites. And at some point we had interest because some of those parasites were not parasites, but they were beneficial for the host. So we were curious about the mutualists, but we never care much about commensals that are the ones that indeed dominate the communities. Then we started to use molecular methods that are not targeted. So we don't necessarily need to look for something. And then we started to observe the diversity of things that are living within animals. And we could report that most of them were indeed non pathogenic and non-beneficial. However, the eukaryote field is still living in this pre-microbiome era. And the reason why is that it's reflected here. So this is a meta-analysis we did for a publication. I forgot to add here the reference in Current Biology in 2017, where we compiled all the genomic information available for bacteria and available for eukaryotes, all the metabolic coding information available for bacteria and available for eukaryotes, and all the metagenomic information available for bacteria and available for eukaryotes. So you can see here, the difference is huge. And in many cases, for example, here, most of the eukaryotic genomes don't belong to microeukaryotes. They belong to multicellular organisms. And with metabolic coding data sets and metagenomic data sets, not all these eukaryotic fraction corresponds either to microeukaryotes. So essentially, we are kind of like starting to explore the diversity of eukaryotes, not only in host-associated environments, but also in free-living environments using molecular tools. If we compare ourselves with scientists working on bacterial diversity. So that's why for microeukaryotes, the framework is still this. Most of them are parasites, some of them are mutualists, and there are some commensals as well. And here, this is the same tree that I used at the very beginning, where I plotted, and we plotted for a review in functional ecology last year, in 2019, all the different parasites, commensals, and mutualists of animals present in the eukaryotic tree of life. And as you can see here, most of them concentrate in three areas of the tree. So the excavates, the alveolates, and the opisthokonts. Now I'm going to show you some examples of organisms within these groups that are parasites, mutualists, and commensals. Let's start with parasites. So you can see here, most of the parasites are within the excavates and within, sorry about that, and within the alveolates. Okay. Within the alveolates, I'm showing you here three examples. Two of them are marine and one is not, but this one is very well known and is the Malassezia Asian. It's an epicomplexan, Plasmodium. There are different species. And most of you probably know about malaria, so I'm not going to go deeper into that. We have also the Perkinsids, the egg cells. They infect shellfish, and they are very important for economic reasons, especially for countries where shellfish is exported in farms. And we have also the Syndinians. Syndinians are a very interesting group related to, well, they are indeed dinoflagellates. So, but they are parasites. They can be parasites of dinoflagellates, and also they can be parasites of copepods. Copepods being one of the most abundant animals on Earth, and they can parasitize also other organisms such as corals. And this group happens to be very abundant as well in the water column because their role as parasites of dinoflagellates. Within the excavates, we have, for example, Trypanosoma brucei that causes the sleeping sickness, and we have Naegleria fowleri, that is the brain-eating amoeba. This is not a marine organism, but a freshwater organism that kills 90% of the people infected by it. And for example, we have it in Florida. So if you go to Central Florida and you want to have a swim in a lake there, better be careful because we have Naegleria there. And what about mutualists? Same story. We have lots of mutualists here within the excavates as well as within the alveolates. Some examples: Symbedium, probably one of the most well-known mutualistic microeukaryotes. It's also called Zooxanthella, and this is an algal symbiont of corals, and it's essential for the survival of reef-forming corals. Currently, because of climate change, reef-forming corals are expelling their symbionts in a process that is named bleaching, that happens when there is a heat wave. In this process called bleaching has this name because corals, they lost their color when they expel the algae. So the algae provides a color to the coral, and when they expel the algae because of heat stress, the coral loses the color. These algae, these dinoflagellates, they go to the water column, and if the conditions go back to normal, they can come back to the coral. But if the conditions don't go back to normal fast enough, the coral will finally die. And that is happening more and more often. So the bleaching events are longer and longer. So corals are dying more and more because they lose their symbiont. Another alveolar symbiont is an apicomplex. And in the previous case, we showed Plasmodium that it's a parasite. Here we have Nephromyces that it's a mutualist of tunicates. It's not super clear what is the role of Nephromyces, but it has been shown that the presence of Nephromyces in the renal sac or of tunicates provides an advantage to the tunicate. So based on that, using the fitness concept, Nephromyces will be a mutualist. We also have mutualist in the excavates. This is an example that is not from marine environment, and those are the termites. And it's well known that in the gut of the termites we have a diverse community of parabasalids and oxymonid excavates that are responsible, together with a bacterial community associated to them, for the degradation of lignin in the wood of termites, of lignin from wood in the gut of termites. And what about the commensals? As I mentioned before, we don't know that much about commensals because we never had the interest, and we were using targeted approaches. And some of the commensals that you see in this tree, they used to be considered parasites, and some of them still are. And there's a debate if they are actual parasites or if they are commensals. There are not many examples from marine environments here that are well known, but I'm going to show you a couple of examples in humans. We have blastocystis that it has been always considered a gut parasite of humans, but recently it has been shown that most of us we have blastocystis in our gut, and when there's a phenomenon of dysbiosis, we lose it. And in some cases, there are some hypotheses that they say that the loss of blastocystis induces this dysbiosis. So blastocystis might not be indeed a parasite, might not be even a commensal, it might even be a mutualist, but there's not enough proof to indicate, to assess that. And then another example is Malassezia. Malassezia is a fungi that lives in our skin, and it causes dandruff, eczema, and psoriasis. So based on that, we will consider this a parasite. But all of us, we have Malassezia in our skin, and it only causes dandruff, eczema, or psoriasis in individuals that are susceptible to have this disease, but not. And it's because of Malassezia, because Malassezia consumes the lipids from your skin, and the byproduct can induce this dandruff, eczema, or psoriasis in individuals that are sensible, but not in most of the people. So what we have here is what I like to call the symbiotic continuum. So we have organisms that are mutualist, we have organisms that are parasites, and we have organisms that are commensals, and we have also organisms somewhere in between. This continuum is not static, it is dynamic, and it does depend on both the organism itself, so the microeukaryote, and also the conditions of the environment, so the host. And an organism that in certain conditions it's a parasite, in other conditions can be a commensal, and organisms that are in certain conditions mutualist can become parasites. And I'm going to show you some examples, one particular example later. So this was an introduction to the whole concept of the eukaryote. And now, I have a final slide. I'm gonna try to go fast with this one. What I'm going to show you here is that some people have been claiming for the last couple of decades that parasites are genomically different and mutualist as well from free-living organisms. And recently it has been shown by sequencing the free-living counterparts of parasites and mutualists that the genomic toolkit that certain organisms use to parasitize or they use to behave as mutualists, it was already present in these organisms, free-living organisms, related free-living organisms that that were not associated with any host. So basically, what has happened is that some of this genetic toolkit that it's already available in a free-living lifestyle can be adapted to the parasitic or to the mutualistic lifestyle, making even more relevant this concept of continuum. Okay. And now I'm going to show you some examples in the marine environment with a particular focus on corals, because it's the marine organisms that I work with mostly. Okay, I don't know if you're familiar with corals. Corals, they form these amazing colonies, and these colonies, they can form reefs. And these colonies, they are formed by these tiny little polyps. But the corals are more than just a colonial animal. So corals cannot live without their associated microorganisms. Indeed, no living being can live without their associated microorganisms. But for corals, that's very characteristic, and it has been very well studied. And among the microbes living within the coral, there are three groups that have been studied deeply. One is the Symbiotium, the zooxanthella that I presented before. So it's a symbiotic algae that is essential for the survival of free-forming corals. Then there is a complex community of bacteria that has proven to be also crucial, and a complex community of viruses that I presume that Forrest Rower was here and probably introduced you to them. Apart from them, there are other microeukaryotes living within corals. Yes, they are, and we know something about them based on microscopical and isolation studies. So we know there are other types of algae, like Ostriobium, that it's chlorophyte. There are apicomplexans, there are ciliates, there are other heterotrophic organisms, such as here we have Labyrintholomycetes, and here we have Neusauropistocon. All these community of microorganisms, together with the coral host, they constitute the coral holobiont, that it's an ecological unity, and in some cases, there is still some debate about it, an evolutionary unit. So based on the techniques that we have been using so far to study coral microeukaryotes, we can identify representative of the groups, of the eukaryotic groups highlighted in orange here. Most of them being fungi, ciliates, and Labyrintholomycetes. Again, the study of the coral eukaryote, it's still at the pre-microbiome era, and most of it has been based on microscopy and not on molecular techniques. I have been doing some analysis using molecular methods to study the coral eukaryote, and I started by scavenging the bacterium. So microbiome studies. Basically, what I did was to use the sequences that were previously characterized as cyanobacteria, as novel groups of cyanobacteria. I reanalyzed them, and what I found is that they were not novel groups of cyanobacteria, but they were the 16S coming from the chloroplast of a group of algae living within the corals, in this case, Ostriobium. And Ostriobium is a very interesting algae. It's an endolytic algae that lives within the skeleton of the coral. And in normal conditions, it remains there. But if there's a bleaching event, this phenomenon where the algal symbiont of the coral leaves the coral, for a certain period of time, Ostriobium moves up. So here you have the living tissue of the coral. So Ostriobium goes from the skeleton to closer to the living tissue of the coral, and it starts providing photosynthetics to the coral and can keep the coral alive when the zooxanthella, when the algal symbiont is not there, allowing a more extended period of time under these bleaching conditions. But if it takes too long for the algal symbiont to come back, Ostriobium will overgrow the coral and will kill it. So Ostriobium can play all the roles across the symbiotic spectrum. So it starts as a commensal, living in the skeleton below the coral tissue. For a certain period of time, can become a mutualist, and if things go really bad, it becomes a parasite and kills a host. We use a similar approach, scavenging data from microbiome studies to study another algal symbiont, or in this case, it was thought to be a symbiont, but we showed that it was not a symbiont. That was Chromera velia. Chromera velia is one of the latest groups of algae described. So it was described in 2008, and it was a completely new group of algae that was at the base of apicomplexans. So this algal group was the ancestor of the group where the malaria parasite belongs to, and most of the apicomplexans are indeed parasites. It was isolated from a coral, and for many years it was considered to be a coral symbiont. But based on our analysis of this 16S microbiome data that allowed us to report the chloroplast of Chromera, we show that indeed Chromera it's not strictly associated to corals, and in most cases you cannot find any signal from Chromera in corals, despite you can find Chromera in the biogenous sediment, that is the sediment below corals. So it lives in the coral reef, it doesn't live within corals. And you will ask why I was using 16S to study microeukaryotes when the barcode that it's commonly used to study microeukaryotes is the 18S of the ribosome. Well, so the reason why is that after the publication of the first human microbiome study, the community working on eukaryotes, they asked for the same type of effort to study microeukaryotes. Here they call it eukaryoton, but then they changed the name because they didn't like it. So that's not on me. Currently we are calling it the eukaryon. Since 2013, there are only 13 papers that have focused on the study of the eukaryon. Maybe I'm missing some, but it's not going to be more than 20 for sure. And why is that? Because if you compare the number of papers studying the microeukaryotes within the microbiome to the number of papers studying bacteria within the microbiome, there are like thousands of papers studying bacteria within animal hosts. Well, the reason is that, as I mentioned before, I mean, we use 16S as a barcode to study bacterial communities in the environment, host-associated or free-living, and we use 18S to study microeukaryotic communities, and it has been used for a long time in free-living environments successfully. But the problem in host-associated environments like animals is that animals are also eukaryotes, and animals also contain the 18S. So when you look at your samples, most of the signal you retrieve corresponds to the host. So you don't see the microorganisms living within the host. In these 13 papers, different strategies have been implemented, like using blocking primers, using primers cocktails, using universal primers. None of them have been very successful, and we came up with an alternative approach that has been very useful to explore this microeukaryotic diversity using an amplicon sequencing strategy, the same one that has been used since 2012 to study the microbiome. So what we did is that we found a set of primers that were designed to study parasites in different types of animals, and we tested them in silico and compared it with other primers that were universal primers for 18S. What we observed for this set of primers, the ones in orange, is that they were very good or as good as all the other primers that have been used so far to study microeukaryotes in free-living environments for most of the microeukaryotic groups, but they were very, very bad retrieving the animal signal. We look at the signal that they were retrieving, and most of it corresponded to porifera. So also some cnidaria, most of them being myxozoans, that it's a type of Cnidarian parasite. But still, they were very, very bad at treating most of the animals except of spongy. So we assumed that we could use them to explore the microeukaryote of animals. So that's what we did. This is a test that we run using these primers. In silico, again, for Cnidarians, for humans, and we observed that they were very, very, very good, but they were not for ctenophores, for example. In the case of Cnidarians and humans, we did a comparison between universal primers and these metazoan excluding primers. And for example, for corals, 80% of the signal came from the host. And when using these non-metazoan primers, only 25% of the signal came from the host. In the case of humans, 99.9% of the signal came from the host. When using these primers, only 20% of the signal came from the host. So this set of primers will allow us to explore the eukaryote in different animals. And we did so for corals. I'm not going to go really deep into it, but using 18S, we could observe the dominance of Symbiodinium, as expected, because it's the algal symbiont of corals, but also the presence of apicomplexans that was unexpected, as well as Syndinians and red algae. By using this strategy, we increased by 5 the known diversity of microeukaryotes associated with corals. You see here the ones I showed you before that were described since recently based on microscopy and isolation. And here are all the groups that we could report using 18S metabar coding. As a practical example, we explore further this apicomplexan signal we observe using 18S because since the 80s there were these reports using microscopy and 16S and 18S of a putative apicomplexan in different corals, but because we were using independent markers and techniques, we couldn't actually assess how this apicomplexan was, how important was this apicomplexan for corals. Using metabar coding, we could determine 18S metabar coding and anti-metazoan primers. We could determine that 70% of the analyzed coral samples, they contain these apicomplexans, and in some cases, up to 50% of the eukaryotic reads, including the algal symbiont Symbiodinium, up to 50% belong to this apicomplexan. I'm gonna go fast because I'm explaining a lot at the very beginning of this talk. So just gonna say that we could access the diversity of this apicomplexan. We could find it in different corals in the wild, or in aquaria corals. As well, using metagenomic data, we could correlate this 18S signal to the 16S signal that has been reported before. So we could link the previous studies where they use 18S to report this apicomplexan with the ones using 16S. And as well, we could do imaging, and we could do FISH for 18S and 16S. We could localize this apicomplexan in the tissue of the coral, and also by doing TEM, we could correlate the morphology of these apicomplexans with previous reports, and we can confidently say that this apicomplexan is the one that has been reported since the 80s using imaging 16S and 18S. Interestingly, this apicomplexan, it's a coccidian. It has is Toxoplasma and Sarcoidosis that are well-known parasites, but because of its epidemiology, because of its presence in 70% of corals, we do not think that it's a parasite. As well, it's an interesting organism because it contains a remnant plastid that all apicomplexans contain, that it's called the apicoplast. And interestingly, it contains all the necessary genes, but 3 to synthesize chlorophyll. And we don't know exactly why, and we don't know exactly how do they use this chlorophyll synthesis operon if it's not for chlorophyll synthesis, or and if it's not for photosynthesis. So that is something that we are still working on it. Another example of the use of this molecular approach to study the eukaryote in a marine animal is the study of the response to thermal stress of Paralysia clavado, the red Gorgonian in the Mediterranean Sea. In this case, what we did was a common garden experiment. So basically, and we had different populations of Gorgonians from different parts of the Mediterranean. Here you'll see 5 different populations. We kept them in the lab, and we had a control and a treatment population. The control population was at 18 degrees Celsius, that it's our standard conditions. And the treatment, in the treatment populations, we increased from 18 to 25 Celsius. That will be a heat wave condition. Based on the results that we obtained, we found that different coral colonies could be classified as resistant, normal, sensible, and sensible. And across these 5 different categories, we compare the eukaryote. This is a work that is being done by Anthony Bonacolta, one of my students at Erasmus. And what we observe here, first of all, we compare the coral with the food we provide to the coral in the water to make sure that there was no cross-contamination and the microkaryotic communities were very different. And then we compare these different sensible to resistant populations. So what we observe is that there was a change on the microkaryotic community, that it's more clear here. So in the sensible colonies, the epicomplexans dominated the coral eukaryote, and then there was a shift towards coelids and, interestingly, towards a group of synthenians that are known to be parasites. So these are preliminary results, but it's very interesting because we are observing this trend where there is a clear shift on the microkaryotic population based on tolerance to heat stress. We have done the same for prokaryotes. I'm going to show it now, but the signal for prokaryotes is not as clear as it is for microkaryotes. I'm going to skip this, and this is just another example of how microkaryotes are everywhere. So we analyzed the presence of microkaryotes in a whole range of members of the zooplankton and the meiofauna. So you can see here everything. I'm not going to go into the details because otherwise I'm going to be late to finish this presentation. But I just want to show you this. So we reported different types of microkaryotes, increasing again the diversity of known microkaryotes within each of these groups. This is different from what we observe for corals, because when we plotted all this information together, most of the differences are organized by the source of by the environment where this zooplankton or meiofauna have been isolated from. So all the organisms living in the sediment, they share the microkaryotic community. As well, the organisms living in the plankton, they have a more similar microkaryotic community. And we presume that these differences are because what we are observing are communities that are being consumed. So we are observing food, because for meiofauna and zooplankton, we cannot process the skin, we cannot process different parts, we process the whole organism. So we are observing microbial communities, microkaryotic communities that are going through the gut or that are colonizing the surface. And in this case, maybe are not that different from one group of meiofauna to another, or from one group of zooplankton to the other, but are more different between those two groups, being zooplanktonic animals living in the water column and the meiofaunal animals living in the sediment. Among the different organisms that we reported in this analysis, Syndyniales, Ichthyosporion, Symbioidinium, and fungi were the most abundant. It's not surprising, some Syndyniales are very abundant everywhere and are known to parasite, for example, copepods. Ichthyosporions are known to live in the surface of amphipods and other invertebrates. And Symbioidinium has been observed to live associated to other microkaryotes. We don't know exactly what Symbioidinium is doing within this meiofauna and zooplankton, but I have to say that these organisms were sampled from the coral reef, and maybe they are consuming Symbiodinium there. And fungi, they have been reported to live on top of zooplankton as well, consuming lipids, and they are also reported to be parasites of algae that might be consumed by this zooplankton and meiofauna as well. So apart from all these conclusions, the message I want to give you, the message I want you to take home is that there's a lot to discover in the microkaryotic world. So if you have interest to study microbiome and you have interest on discovering new organisms or new relationships, new interactions, the eukaryote is your place, because there's so few things done that there's a lot of field to run and to explore. So I will encourage you to pursue that, because it's really exciting, because everything you look at it, you observe something new, and that's very exciting. I would like to say thank you to my students and also my collaborators, and I would like to thank you for your attention. And I think I'm a bit late, but